Let's go ahead and get these notes done. I'm going to talk about two types of transports, either active or passive. Passive transport does not require energy, as you can see here, so there's no ATP needed for this. It works just against the concentration gradient, which you know from all your other experiences with concentration gradient. When something is in high concentration, it flows to an area of low concentration so that equilibrium is released between the two concentrated areas. And that's how this whole principle works. You can see I've got molecules here of high concentration. There's lots of little blue balls representing the molecules here. Not very many over here, so they're going to flow across this is the phospholipid bilayer. You see the head and the tail region of the phospholipid bilayer. It's going to flow across there to this area of low concentration until there's equal amounts of molecules on both sides. Okay. Two types of passive transport are diffusion and osmosis. Well, osmosis is diffusion. It is just diffusion that only focuses on water molecules. So diffusion is any kind of molecule. This could be a potassium. This could be a carbon dioxide gas. It could be an oxygen gas. It could be a sodium chloride. It could be anything. And that's what diffusion is referring to, any molecule moving from an area of high concentration to low concentration. Osmosis is the same thing, only it's just water molecules moving from an area of high concentration to an area of low concentration. And they'll likely move, in this case that we're talking about, across a cell membrane, which is semi-permeable. So it's going to let some things across, and in other things it's not going to let cross. You probably saw this somewhere. I'm sure Bill Nye did it in one of his videos that you watched in middle school or elementary school. And it was basically he put a drop of food coloring into a beaker. At first there was a high concentration of the green food coloring in wherever the droplet landed, and then after 5 minutes all the color evenly dispersed throughout the entire beaker. That is moving from an area of high concentration to an area of low concentration. The next slide is talking about how cells are going to respond in these different environments where there's different concentrations and different mixtures and so forth. So we're going to look at three solutions: an isotonic, a hypertonic, and a hypotonic. And you can see I've got my three pictures here depicting that. And in these solutions, cells will behave in very predictable ways. For instance, in an isotonic situation, iso meaning equal, there's an equal amount of water, and let's just say I'm going to throw a molecule out there like sodium, inside the red blood cell and outside the red blood cell. That's what this is right here. This is a perfect red blood cell. So because there's equal concentration gradient, the molecules are going to keep moving freely across the membrane, and there's no area of high concentration versus low concentration. It's all balanced, equal, homeostasis. Now, in a situation where you have more solute in the exterior environment of the cell, for instance, this scenario, it's a high concentration of sodium in the bloodstream. Maybe you ate something that was really salty, and it put a high concentration of sodium into your blood cell or into your bloodstream. In this case, the cell is actually going to shrivel, and the reason it does that is because you've got water on both sides of the membrane. If you've got more salt out here and less in here, the water is actually going to move out to this higher sodium area to dilute that high concentration of sodium chloride. So you can see it moves from an area where it needs to dilute a high concentration. Okay. If you remember, here's my little trick here. If you remember hypertonic and think of hyperactivity, you get all jacked up on sugar and caffeine, and you're just go for about 30 minutes, and then what happens? You just kind of crash and burn, right? You just want to shrivel up on the couch and do nothing. So that's kind of what the cell is. They're hyperactive for a period of time and go, and then they shrivel. And that's how the cell responds in a hypertonic situation. Now, the opposite is true if it's in a hypotonic solution. Hypo, and I use the analogy here of the hippo, okay, because what kind of animal is a hippo? It's a large animal. So in this case, the blood cell is going to get larger. It's going to inflate and bloat. And why does it do that? Well, here again, you've got your sodium concentration and your water concentration, and it's different between the inside the cell environment and the outside cell environment, like your blood plasma. So what happens is you've got less solute out here. Okay, less sodium here, more sodium here. So water is going to flow in to try and dilute that high concentration of sodium, okay, to kind of equal it out. And in doing so, you get a higher amount of water inside the cell, which causes it to bloat. Okay, so that's how using the principles of passive transport across a membrane, how blood cells would respond to that. All right, so the next one, still passive, but with some assistance, and that's why we say facilitated diffusion. And what the assistance is, there's no energy needed. It's just an open channel. Okay, so someplace larger for the molecules to get through that maybe are too big to pass through the membrane directly by the concentration gradient. So you can still see it's flowing according to the concentration gradient. They just can't pass directly across the membrane. So they pass through these little tunnel areas in the cell membrane, which we actually call protein channels. Okay, so here's one, in this case, this molecule can't cross the membrane directly. It passes through the tunnel to get to the area of low concentration. Still diffusion, just needs a little helper with the protein channel. No energy. Now we're going to talk about energy required. This is active transport. Requires energy in order to get the molecules pushed against the concentration gradient sometimes in order to move molecules across the membrane. And I'm going to show you an animation on this tomorrow, but to get the gist of it, active transport requires energy, and the energy that we're talking about is ATP, which stands for adenosine triphosphate. Okay, and what happens is, if you look at the scenario here, molecules are inside the cell membrane. They come into this pumping station. These are like pumping molecules now, this purple stuff, and they load in here. The pump is actually going to change itself. It's going to reconfigure, and you can see how it was open inside and now it's open outside. So it conforms, it changes. And the animation that I'm going to show you is going to make it a lot clearer in your mind. The bottom line is it takes energy in order for this pump, this purple representation here, to transform itself so that it moves from the inside the cell to outside the cell. So that's why it said it requires energy. And it's still trying to maintain that homeostasis most of the time. All right. Specific types of active transport that you need to know about. Endocytosis and exocytosis are the two general category words. And in endocytosis, endo as a prefix means in, so we're talking about molecules or substances coming into the cell. You can see here's the open environment. The cell membrane just kind of puckers a little bit, divots in. The substrate, whatever it is, goes into that opening, and then it's sealed off. The cell membrane just reconforms around it, so it becomes like a vesicle encapsulated inside the cell now. And then the membrane will start to dissolve away. These phospholipids will go back and join the cell membrane, and whatever that substrate is now inside the cell. Two specific types of endocytosis are phagocytosis and pinocytosis. Phagocytosis you mostly see when you're talking about pathogens, foreign substances, or bacteria, these solids that need to be moved around or gotten rid of in your body. And one thing is your white blood cells will come in and they will engulf it and take it in and then destroy it. So that's kind of how they work in a general speaking. But it's removing bacteria or pathogens. That's what phagocytosis is specifically talking about. Pinocytosis is that same movement inward, is something into the cell, but it's specifically looking at fluids, so like liquid type substances that come in. So same process as you've seen here, only we're referring specifically to fluids. Exocytosis, which is the other general category. I don't have any specifics that you need to learn for this one, but we're talking about getting things out of the cell. So here you can see the cell membrane encapsulated whatever was inside that needs to get out. And then the cell, it becomes part of the cell wall. It merges, and they kind of all become one, and then it opens up to the exterior and lets whatever was inside now out. Okay? And that's exocytosis. And think about your prefixes. Exo meaning exit, right? All right, well, that's all I got for you. Quick little notes. Hopefully that wasn't too painful. Take your notes, and I will see you in class tomorrow where we will go through and do that quiz, and I'll show you an animation of this. If you can't wait, there's lots of stuff on YouTube. If you just Google active transport, you will see how it works. Otherwise, I will show you it tomorrow. All right, enjoy.